So thank you so much for having me. I'm really excited to be here. Obviously, if you've met scientists, we love talking about our science, and we love even more talking to non-scientists about our science, because everything that I do and everything that I focus on is in the pursuit of knowledge. And so what I wanted to do is start off a little bit with where my philosophy of science comes from. So I figured a good place to start was with Descartes, right? So Descartes says, I think, therefore I am. And this is a fundamental principle in how we view neuroscience, because we're thinking and therefore we are. So therefore we should study neuroscience. It's who we are. I would tweak this statement though, through my education. I believe actually that I perceive, therefore I am. And I say that because by thinking and being means that it's all in here. There's nothing in the physical world. But perceiving and perception is what influences how it is that we think. And this is called sensory neurobiology. And so here we have this beautiful food illustration that elicits all of these different senses. So you may look at this and see the bird, and maybe you hear the bird that you heard this morning, or maybe you haven't quite eaten your pizza yet, and you're like, maybe pasta and beans would have been the way I wanted to go. And you're thinking of the food, or maybe you're drawn to the color and the visual aspect of this. There are all these senses together that create some kind of thought in your mind as to what it is that you're feeling. And so I pursued my undergraduate degree at UC San Diego. So at UC San Diego, I did a dual degree in neuroscience and in psychology. So my focus was really on what is the circuitry and what is the perception and what is the interface between circuitry and perception. And so I like to show this great visual illusion. So as you're looking at this, you're probably thinking, why are these dots moving all over the place? What is that black dot? I can't quite get it into focus. This visual illusion is a direct result of the circuitry in your retina that is initially filtering the visual information that's coming in. So what this visual illusion is really showing you is that this is not true to the color and the physical stimulus that's here. What this actually is a reflection of the circuitry of feeding the light stimuli into our mind. Oh, there we go. And so I especially appreciate this because in college I rowed. And so as I'm there and I'm rowing and my body is hurting, I have to think to myself, this is an illusion. I need to just keep on going and keep on rowing. And so this is a practical aspect of how I navigate my life. So from undergraduate, I went on to Yale, where I got my PhD in cellular and molecular physiology. And I shifted a little bit from the obvious senses to one that wasn't quite so obvious, which is the visceral sense. And so by the visceral sense, what I focus on is the vagus nerve. This is my favorite nerve because the origin of it is the wandering nerve. So this nerve connects the brain into the viscera, and it kind of just wanders around throughout all of our different organs and innervates everything. And so it innervates things like the gut, it innervates the lungs, it innervates the heart, and then all of the cell bodies of these neurons of the vagus nerve come into what's known as the nodose ganglia. And they're all dispersed within there. And so in my graduate work, what I focused on is how is it that if I want to study the lung, which is what I did for my graduate work, how do I study the nodose and only get lung innervating neurons? And what it became apparent is when you study the vagus nerve, you've got to go to the organ itself, because all of these collection of neurons, it might be tempting to go there, but they innervate everything. So you have to be able to focus in on just the neurons that you're interested in. And so from my graduate degree, I went on to Duke University, where I am now, and this is where I did my postdoctoral work. And this is where I got into the sense that I love the most, which is the gut sense. And so the gut sense is how it is that this gut is telling us information about the food that we're eating. So let me just orient you a little bit and the physiology of it. So I study how it is that the gut and the brain connect. This is specifically through this vagal nerve connection. Within the gut lumen, we have multiple villi. Within a single villus, you'll see there's a layer of epithelial cells on the surface. And there are multiple types of epithelial cells. The one that I'm interested in is the neuropod cell. This is the sensory cell of the gut. And this cell is able to detect things in the lumen, such as mechanical stimuli, thermal, or chemical, which is basically nutrients. It takes this information and then it turns it into an electrical pulse that then communicates directly to the nerve. And in this way, this is how we take the physical stimuli of what's in our gut and we turn it into an electrical signal that the brain can understand. And so this transduction is what it's called, allows us to now see what is our gut doing and communicate that very rapidly to the brain. So when we discovered this sense in 2018, that there's this direct connection, the number one question I got is, what is a gut sense? What could our gut possibly be telling us that's more or less or different than what our tongue or our nose is telling us? Well, there's this interesting phenomenon that we've seen over and over again in many animal models. And it's this question of artificial sweetener versus caloric sugar. And so we have artificial sweetener on the market, and everyone thought artificial sweetener is here, obesity is going to be cured, and yet it didn't work. We still love our caloric food. We still don't particularly love artificial sweetener. And a number of people have done this study, and what we find is that, so this is a mouse, this is mouse work, where we give the mouse two options. It has two bottles. One of them has sucrose, which is caloric table sugar. And then the other bottle has sucralose. This is what's in sweetened loads, an artificial sweetener. And so over here on the right, we have time on the x-axis, and on the y-axis, we have cumulative intake. So what we see is that the mouse very quickly discovers which solution has caloric sugar and then solely drinks from the caloric sugar over the artificial sweetener. And what people have shown is that this distinction is independent of taste in the mouth. In fact, you can knock out sensing in the mouth and you still get this phenotype. You can bypass the mouth completely and inject the nutrient directly into the intestine and you still get this phenotype. So there's something in the gut that is allowing us to tell the difference between these two solutions. And so in the laboratory, we use a number of different techniques. And one of the main techniques that we use is called vagal cuff recording. And so in this technique, so I mentioned neurons communicate via electricity. So what I have is I have two electrodes around the cervical vagus, around the vagus nerve, and it's measuring the differential electricity between those two electrodes. And then I can perfuse nutrients through the lumen of the intestine. And using this, I can tell, based on the nutrient that I perfuse, is there a neuronal response? Does the neuron care about the nutrient that I'm perfusing through the intestine? And so here's an example of what these recordings look like. Again, on the x-axis, we have time. The dark bar is when I perfuse in the stimulus. And on the y-axis, we have the normalized firing rate. And so there's always this baseline activity. So we're normalizing the baseline activity. And what we see is when I perfuse in sugar or sucrose, there's this rapid response on the vagus nerve, indicating that something in the gut is telling the vagus nerve, this is sugar, it's good, you should eat more. And so we tested a number of different types of sugar. So sucrose is a disaccharide. It's made up of glucose and fructose. Interestingly, the vagus nerve does not care about fructose. And there's some work that has suggested that actually the spinal cord cares more about fructose than the vagus nerve. So there are multiple circuits going on here. We tested some sugar analogs. So these are things that mimic sugar but are not sugar. And then we tested a number of artificial sweeteners. And we see that actually the vagus nerve is responding to all of these. So you're like, okay, well, if the vagus nerve is responding to all of these, like, how's it telling the difference? So we go to the gut epithelial layer, to these sensory neuropod cells. And I ask the question, does one neuropod cell respond to both caloric sugar and artificial sweetener, or are there different populations responding to different stimuli? And so here I'm measuring calcium activity. This is a measurement of activity in these cells. And what we're seeing down here is the overlap. So I exposed these cells to both either glucose, then sucralose, or vice versa to make sure there's no masking effect. What we found is that the majority of them really like the caloric sugar. They like glucose. That wasn't surprising. There's also a small population that likes the artificial sweetener, but doesn't respond to glucose. So that's interesting. And then there's another population that can't differentiate between the two and just responds. And so this gets us into the question, okay, so these cells can tell the difference. So the vagus nerve can't really tell the difference, but these cells can tell the difference. And so the question is, are these cells the ones that are transducing it? Maybe it's some secondary messenger, maybe it's another cell. And so in order to ask this question, we turn to optogenetics. So optogenetics allows us to use light in order to block the activity of a cell. And so this is using something called Kaelberer. What happens is that this 532 nanometer light, it's green light, when it shines onto this channel, what it does is it inhibits the cell's ability to act. So now nothing that comes in will create a response. So if we go back to our vagal nerve recording, what we see is, so the dark line on the top is sucrose. It's their classic, it's got a great response. Then there's the dashed line below. This is when we optically inhibit neuropod cells, and it completely abolishes the response. So this is indicating these neuropod cells are actually necessary in order for the nutrient to be transduced. And if we look at artificial sweetener, we see the exact same thing. So now we're getting a little closer. Okay, these neuropod cells, we know that on the individual level, they can tell the difference. We know that they're required to transduce onto the vagus nerve. And so how are they telling the difference? So in order to get to this question, we wanted to look at what are the receptors that are sensing this? Because as far as we know in the tongue, there are sweet taste receptors, and sweet taste receptors do not distinguish between artificial sweetener and sugar. Everything that's sweet tastes sweet to them. So we used pharmacology. We're using this, this drug called Coumarin to block sweet taste receptors. And in the gut, there's something else that we have there because we need to absorb sugar. We absorb sugar using a transporter, the sodium glucose transporter. And so we thought maybe this is sensing the sugar. So again, we go back to vagal cuff. And this is now a lot of lines on top of each other. So the green one, the short dotted line, oh yeah, I gotta point this way. I'm gonna do this. The short dotted line is going to be blocking just sweet taste receptors. And I can highlight just the top, and we see that doesn't actually affect the sucrose. So it's the caloric sugar doesn't seem to be affected by that. The fluoridazine, which is blocking this transporter, is this line now down here. So that completely abolishes the response to caloric sugar. So we see for sucrose, it seems the transporter is required, the sweet taste receptor is not. And we saw the exact opposite for artificial sweetener, which is that what we see here is that the sweet taste receptor blocker, Coumarin, is down here, yeah, the dotted line, completely blocks the sucralose response, whereas the transporter blocking the transporter has no effect on the sucralose response. And so now we're starting to paint this picture of, these cells are telling the difference between artificial sweetener and caloric sugar because artificial sweetener cannot be transported into the cell and caloric sugar can. So it's using these two different receptors to distinguish between these two molecules. And so when we do vagal nerve reporting, the nerve is a trunk. The nerve is all of the neurons of the vagus nerve being recorded at the same time. So we know that these cells, the neuropod cells, are telling the difference, but we see a general global response. And so the question becomes, are the individual neurons responding to both of them, or are they responding to different ones? And so to look at this, what I did is calcium imaging of the nodose ganglion itself. So now I can see single neuron resolution and then again, perfuse the different nutrients through the small intestine. And so when I do this, I did sucrose and then sucralose. And so here we just have pseudo-colored neurons that responded to sucrose, neurons that responded to sucralose, and then the merged image. And what we found is here, it's now a graphical representation. The neurons that were responding to sucrose were not responding to sucralose. And the few that responded to sucralose were not responding to sucrose. So this is indicating that there's actually these parallel pathways that are communicating one solution versus another solution. And so then the next question is, well, what about the signaling molecule, right? So we have the sense, whenever I think about neuropod cells, I think like my head is on the apical membrane, and I think that my arm is out touching the neurons. So I always talk about it in that way. But yeah, so we have the different receptors, but then I'm signaling something to the vagus nerve. And so we want to know what is the neurotransmitter? What is that communication molecule? And so one of the first things we looked at is glutamate. We knew that glutamate got released in response to sugar, but is it responsible for this communication with the vagus nerve? So here, these glutamate blockers, they are shorthanded KAAP3 because they have really long and complicated chemical names. And so what we see, again, we have the normal sucrose response. And then when we block glutamate receptors, this is this dotted line down here, we see that that response has been attenuated quite a bit. So we know, so sugar is at least eliciting a glutamate response or a glutamate signal onto the vagal nerve. Sucralose, however, glutamate had no effect on the response whatsoever. So now we're seeing that not only was it different receptors that were being activated, but it appears to be different neurotransmitters that are being released. And in fact, we went on to show that sucralose seems to be eliciting ATP release. So now we have two different receptors. They're releasing two different molecules, and then the connection is onto two different populations of neurons. So now we have these two parallel circuits in the gut that are actually able to differentiate between these two solutions. And so now we wanted to do this. All right, this has all been in a dish so far, in an anesthetized animal. What about that animal that's awake and moving around and able to consume solutions? Well, in order to do this, we had to bring optogenetics to the gut. And so one issue with optogenetics is that it was developed for the brain. In the brain, you have this skull that you can attach anything to, and your brain's pretty held steady in there. Your gut, however, moves around. You've got muscles that are moving all over the place. And so these stiff silica fibers that are generally used for optogenetics puncture the gut. So what we have here is on the left, we have the stiff silica fiber. There's a little bit of a gelatin cover, which is about the consistency of the gut on this jar. On the right, what we have is this flexible fiber optic. So we teamed up with the scientists at MIT, material scientists, that were developing this flexible fiber optic. And we said, well, we have a use for this. This should go in the gut. And so what we'll see here is turn on the fiber. If we lower now the fiber down, you can see the stiff silica fiber punctures our artificial gut here completely. And the flexible fiber optic just bends and is able to move. And so we developed this flexible fiber optic to be able to implant it into the gut of the mouse. And so here we see it shining within the duodenum, so the proximal small intestine of the mouse. And so now these mice, they get this implant, they move around, they can eat fine, they go about their lives and they're happy. And so we want to know, well, does this have an effect on the consumption of sucrose versus sucralose? So here we have just the control laser. So this is the wrong wavelength laser. And we see that it's true, the mice still prefer sucrose over sucralose. But when we optically inhibit neuropod cells, the mouse is no longer able to tell the difference between sucrose and sucralose. And in fact, the mouse is consuming the same amount of solution. It's now just consuming equally from the two different bottles. And so we have basically, this experiment, we've temporarily blinded the mouse to which nutrient has caloric value and which one does not. And I should say, if we were to now go and do this experiment again without the silencing, the mouse still prefers sucrose. We have not gotten rid of this memory. It's just at this moment, we blinded the mouse at the point that it's critical. And so these experiments have shown that the gut actually seems to be doing a lot more than we realize. And in fact, it's driving our choices as to what it is that we are eating and what we're not. And the way that I think about this is that what I think is when you eat your food, you sit there and you're like, that was really satisfying. That was a gratifying meal. I really enjoyed it. Or sometimes you eat your food and you're like, that didn't really hit the spot. Like, let me eat a little bit more of this or a little bit more of that. I think that feeling is our gut sense. That is the sense that the gut is communicating, saying, yes, you should eat this. No, you should not eat this. And so the next time that you're choosing your pizza, it's probably your gut that's saying, did you really want pepperoni? Or maybe you want pepperoni, sausage, and chicken. And so that's just a little bit of a slice of what it is that I do in the laboratory. And I'm happy to take any questions. Thanks for your time.
Yeah, actually. Thanks very much. We actually had Dr. Borges here a couple of years ago to give a talk, so it was fun to hear this update. But yeah, so I'm still struggling with, and you sort of gave your impression of how it's influencing our behavior, but can you talk about specific regions? Is it amygdala, or what is it that's actually driving our behavior in this unconscious way?
Yeah, so I mean, that's a loaded question, because I would say that let's say visually, that's a visual example is, let's say I prefer green shirts over red shirts. Okay. You give me the option of a green shirt and a red shirt. I will prefer the green shirt. But preferring the green shirt and liking the green shirt just means I have to know that it's green, and then comes in, there's some kind of a dopamine reward saying, yes, I like green. Or there's like a little bit of serotonin sometimes, and you go and you pick up that green shirt. And so there's a whole circuit involved in that. So if you're asking where the memory of liking the green shirt is, the memory is actually encoded in the entire circuit. Because if you didn't have photoreceptors, would you like the green shirt? Like, because you can't tell the difference. And so that aspect of it ends up being a little bit of everywhere, right? So we know that encoding in the memory is more in the hippocampal region. We know the emotional value is attached to the amygdala. If you trace the circuitry from the gut, we're going up into the brain, and actually the first place we hit is the feeding centers.
Okay. So the glutamate in the gut doesn't.
The glutamate in the gut is just in the gut. That's just at the synapse, right in the gut. Mm-hmm. Yeah. But yeah, and I would also say that for the behavioral experiments, what we did a behavioral experiment where we then went in and instead of using light to block just the neuropod cells, we used glutamate receptor blockers, had the exact same effect. Oh, go back there.
How do you see this potentially translating to chronic diseases like diabetes or obesity or other things?
Yeah, and this is a great question. We're understanding more and more about what it is that the desire to eat comes from. And I will say we also, so everything that I showed you was in mouse, but the gut is this really cool thing where we can make human organoids. We can take scrapes of the epithelial layer and we can grow them in the lab, and now we can test. What we tested using this assay is we tested the release of glutamate. This was the obvious question. And we found for humans, it is true as well, that glutamate is only released to caloric sugar and not for artificial sweetener. And so you can imagine that if we know what those signaling molecules are, now we can design our food a little bit differently so that at least that immediate response is there. So you can imagine now having a low calorie option that gives us that immediate glutamate signaling so that it kind of tricks your gut a little bit. Though tricking your gut is incredibly difficult, because I would also anticipate that if we were to chronically silence these cells, there would be a secondary system that would kick in that would allow us to now pick up glucose in, say, the portal vein. That's another strong glucose sensing area. And so for the applications for these things, right, it's going to be multi-level, but this is at least a first step for how it is that we can, at least in the short term, jolt the system. Because I think when it comes to things like obesity and diabetes, these things, some of the more effective things are just jolting the system or like resetting the system a little bit. And then it takes the hard work that we all really don't like of going to the gym and eating better. But at least kickstarting it, we can do it using the system.
When you take these scrapes and throw them in the lab,
Yeah.
presumably there's some gut biome population. Are there any metabolites from our gut biome that could be?
Well, we wash the biome away before we grow them.
Maya, the people online have asked if you could repeat the question.
Oh, yeah.
The ones for the audience, so.
Yeah, the question is when we get these samples from human, are there still bits of the microbiome? And yeah, and the answer is we have to wash them, otherwise they won't grow. The bacteria likes our culture media far more than the epithelial cells do.
Do we have more questions? Go ahead.
How studying this changed like your behavior?
Yeah, so the question is, how has this changed my behavior? I like to think it's changed it for the better. I've decided I no longer feel guilty about eating food that I like. And I think that there's a little bit of wisdom in that, because what I'm learning about how this works and how I think about I perceive, therefore I am, is that the wanting is so strong that you'll overcompensate. People have shown this with artificial sweeteners, that when you have that Diet Coke, you have a tendency then to eat more food because you're almost compensating for the fact that you didn't get the calorie signal. And so I would say that how it has changed is that I don't pay quite as much attention to my calorie count. And instead, I just think of maybe eating a little bit less. Like, sure, I can have some chocolate. I'm just not going to have a ton of it. Like, that's how it's actually affected my eating behavior. I liked how you nodded and then took a bite of your peanut. Exactly. Yeah.
This question might be a little bit outside of your focus specifically, but I was wondering if you've read anything about like neurodegenerative diseases like Huntington's and Alzheimer's, where the patients just crave that extra sugar. I wonder if there's anything with the vagus nerve and your research that might connect with that.
Yeah, I have tangentially been following this because I think it's fascinating thing of whether or not in those degenerative disorders, because a lot of people think that with Alzheimer's, especially, the increased consumption is because they forgot that they ate. But nobody's quite looking at whether or not the signaling pathway into the hypothalamus from the gut is actually being degraded as well. So you're not quite getting that same rewarding feeling that you used to get from eating the food. Then there are other disorders that are more tightly linked to the gut-brain connection. Parkinson's is one of them. So now we've seen that these aggregates of alpha-synuclein, they start in the gut, and you can see them in the gut years before they move into the brain. And there's a researcher at Duke who has shown that from these neuropod cells, you can see the accumulation, and you can see those alpha-synucleins jump onto the vagal neuron. And so this could actually be a pathway of getting some of those aggregates directly into the brain.
So they migrate along the vagus?
Yeah, because once it gets into the cell, there's all this transportation molecules that help move things up and down. And since the vagus nerve, it's bypassing the blood-brain barrier. It's a nerve that part of the cell is down in your gut, the other part is up in your brainstem.
If we don't have any other questions.